Hello everyone and welcome to today's session. And today we'll be discussing the theory related to chapter Principle of Inheritance and Variation. And in today's session we'll be discussing the NCERT sections 4.3, 4.4 and 4.5. And this is the part 5 of the lecture. Before starting this lecture, just want to give you one information that the images, text and other materials used in the presentations of education purposes only. We respect the intellectual property rights of the original creators and owners. If you're the owner and have any concerns or issues, please contact us at sathee@iitk.ac.in and we shall remove the content immediately. Please note that parts of the content may have been generated using generative AI, which is experimental. So what are the objectives of today's session? So we'll talk about linkage and recombination. Okay. Under this topic, we will understand the concept of linked genes and how they intend to be inherited together during the gamete formation. So we will understand what are linked genes and how they are inherited. Okay. Then, with the help of Morgan's experiment, so Morgan conducted experiments on Drosophila. So we will understand Morgan experiments which he conducted on Drosophila, and by his experiments, he coined the term linkage and recombination. So we'll discuss Morgan's experiments as well. Then we'll discuss polygenic inheritance. So in this, we'll be studying what is polygenic inheritance and how the multiple genes contribute to a single phenotypic trait, means how multiple genes control a single character or single phenotype. We'll also analyze how continuous variation in traits like human height and skin color. The human height and skin color, they show continuous variation, means they show a range of phenotypes. Okay. So we will analyze how continuous variation occur in traits such as height and skin color, and how this continuous variation arise due to combined effects of the multiple genes. So in polygenic inheritance, multiple genes are involved, and there is a combined effect of this multiple genes. So we'll study this as well. Then we'll talk about pleiotropy. Within this topic, first of all, we will understand the definition of pleiotropy and how a single gene affects multiple unrelated phenotypic traits. So we'll understand this with the help of an example. So we will understand pleiotropy with an example of phenylketonuria, which is a disease in humans. It is a metabolic disorder in the humans. And what are the phenotypic traits, or what are the phenotypes that you observed in the phenylketonuria in the organism? So today we'll be talking about linkage and recombination, which is under the section inheritance of two genes. We'll talk about the polygenic inheritance. We'll talk about the pleiotropy. And we will summarize this entire lecture. We'll discuss some NCERT based questions related to these topics. In the end, we'll discuss some keynotes that will help you memorize all these concepts, all these topics. Okay, and in the end, there'll be some interesting questions for you. These interesting questions will enhance your critical thinking ability. So let's start with this first topic, linkage and recombination. Now, linkage and recombination was discovered by Morgan. So he coined the term linkage and recombination. So what Morgan did? Morgan conducted dihybrid crosses in Drosophila. Like Mendel conducted dihybrid crosses in pea plant, similarly Morgan also conducted dihybrid crosses in the fruit fly, which is Drosophila melanogaster. And why he conducted these crosses? To study the sex-linked genes. So what was he studying? He was studying sex-linked genes in the Drosophila. Now you can see this is the image showing you Drosophila. Image A, this is of male Drosophila. Image B, this is of female Drosophila. And we have already studied why Drosophila is used in research, why it is a model organism. Now, these dihybrid crosses were comparable to Mendel's dihybrid crosses in the pea plant. So, like Mendel conducted dihybrid crosses in the pea plant, similarly, Morgan also conducted his dihybrid crosses in the Drosophila. So, what he did? He crossed a yellow bodied and white eyed female. So, here he has taken two characters. Okay, the body color, yellow bodied, and the color of the eyes. Okay, what is a dihybrid cross? A cross that involves, you can say, a cross between organism, cross between individuals that differ in two traits. And these two traits are controlled by two genes. So he crossed yellow-bodied, white-eyed females with brown-bodied and red-eye males. And then intercrossed their F1 progeny as well. Okay. Now, what he observed? He observed that these genes that did not show independent assortment. Usually what happens? The genes, they assort independently. There is independent assortment of the genes. This law was given by Mendel. And he proved this law. He gave this law. He explained this law with the help of dihybrid crosses in the pea plant. But here, Morgan didn't observe independent segregation of the genes. And the F2 ratio significantly deviated from the expected 9:3:3:1 ratio that occurs with independent genes. So usually what happens in the F2 generation, the phenotypic ratio is 9:3:3:1. This happens when the genes are independently assorting. Okay, but here, the genes were not independently segregating. And so the phenotypic ratio in the F2 was not 9:3:3:1. The phenotypic ratio significantly deviated from the expected 9:3:3:1 ratio. Now, why was this happening? Okay, so this was due to linkage of the genes. Clear? So Morgan and his team, they conducted dihybrid crosses in the Drosophila, and they discovered genes on the X chromosomes. Okay. Now they observed that in dihybrid crosses where genes were on the same chromosome. Okay. So Morgan, they were studying the genes on the X chromosome. And what they observed? They observed that in the dihybrid crosses involving this X chromosome, okay, here the genes were present on the same chromosome. And the parental gene combination occurred more frequently as compared to the recombinants. So thing here to note is that, first of all, the genes were located on the same chromosome. Okay, not on different chromosome. Genes are located on same chromosome here. And more parental gene combinations occurred. So parental gene combination were more as compared to the recombinants. Or you can say non-recombinants. Non-recombinants means parents only. They were more as compared to recombinants. Clear? So this was because of the linkage of the genes. So you can see here, this A and B gene, they are present on the, you can say same chromosome, and A and B are linked. And A and C, they are not linked. A and C, they are present on different chromosomes. So when the genes are present on the same chromosome, then only they show linkage. Okay, when they are on the same chromosome and distance between the two genes is less. But when the genes are present on different chromosome, like A and C, these are different chromosomes, then these two genes are not linked. So in the case when the genes are linked, more parental gene combination will be observed and less recombinant will be observed. When the genes are not linked, then recombinants will be more as compared to parental. So what were the key findings of Morgan's and Morgan's team. So they said that the parental gene combination appear more, okay, as compared to the non-parental types. Parental gene combinations are more as compared to recombinant types. So this observation helped establish the concept of genetic linkage on the chromosome. So when the genes are linked together, then parental combinations are more. So when the genes are linked together, then they are inherited together. That's why parental combinations are more. Okay. And as they are, you can say, present on the same chromosome and distance between the two genes is less, means these two genes are physically associated with each other. Then the recombination between these two genes is less. That's why parental combinations are more. These two genes are inherited together. Okay, now you can see here, this is showing you linkage and crossing over. So these two genes A and B, they are present on the same chromosome. And the distance between these two genes is very less. These genes are physically associated with each other. Now this C gene is far apart. Okay, now recombination will occur. So A and B, these are being inherited together. You can see A and B, the distance between A and B is very less. They are present on the same chromosome. So they are being inherited together. But you can see recombination is occurring between this. Crossing over is happening here. Exchange of segments can occur between A and C here. Okay, A and B, they are inherited together. So A and B are linked here. Now let's understand linkage and recombination. So what Morgan said, Morgan linked the co-inheritance of the genes. What do you mean by co-inheritance of the genes? Means the two genes A and B, as shown in the figure, these A and B were inherited together. So co-inheritance of the genes. Okay, so Morgan linked co-inheritance of the genes to the physical location on the chromosomes. Okay, physical location where they are present on the chromosome. Okay, so they should be present on the same chromosome and the distance between two genes should be less. Morgan, he defined linkage. So he discovered linkage. He coined the term linkage. He defined linkage as physical association of genes on the chromosomes. Okay, means the genes are linked together. They are very close to each other. Morgan also coined the term recombination. Okay, so he introduced recombination as form of non-parental gene combination. So after recombination, what happens? Recombinants are formed, means non-parental gene combinations are formed. Okay, when the genes are linked, parental combinations are formed. When recombination occurs, then non-parental gene combination occurs. So you can see in this image, two crosses are shown to you, cross A and cross B. Okay, so you can see in cross A, parents are given to you and the genes are also given to you. Okay, Y and W, Y dash and W dash. Okay, clear? So here the positive sign in the superscript is shown to you, Y positive and W positive. Clear? These are the two genes here, Y and W. So you can see here, then F1 generation is given to you and F2 generation is given to you. In the F2 generation, you can see they have shown you parental and recombinant. Then cross 2 also, cross B also, they have shown you parental and recombinant. So in cross A, the parental types are more, 98.7%, recombinant are 1.3% only. In cross B, parental combination are 62.8%, recombinant are 37.2. So in cross A, you can say that the genes are linked together. because you are getting more parental combinations here. Recombinants are less. In cross B, you can say that the genes are, you can say genes are not linked, or you can say they are not completely linked, because still you are getting parental combinations, but recombinants are more. So in cross A, the genes are almost completely linked because you are getting 98.7% parental. So there are two types of linkage: complete linkage and incomplete linkage. In complete linkage, you get almost all the parental combinations only when the genes are completely linked because they are inherited together. So when the genes are completely linked, you will get only the parental type, no recombinant type. So in cross A, you are getting almost 98.7% parental type. So this is, you can say, a type of complete linkage. Cross B is incomplete linkage because although you are still getting more parental combination, but you are getting 37.8, 37.2% of recombinant as well. Okay. Now, Morgan and his group discovered that even when genes reside on the same chromosome, some genes are tightly linked where they exhibit very low recombination, as in the case of cross A that we have seen. And some genes are loosely linked. So there can be complete linkage or there can be incomplete linkage. Or in other words, you can say the genes can be tightly linked or genes can be loosely linked. Tightly linked genes can be seen in this cross A, and loosely linked gene can be seen in cross B. Okay. So when the genes are tightly linked, you will get parental combination. When the genes are loosely linked, you will get recombinant types that will be more as compared to this cross A. Okay, so tightly linked genes, they exhibit very low recombination. Loosely linked, they display higher recombination. And you can see in this image also, when you see the parents, in cross A, the genes are closely situated to each other. The distance between the Y and W gene is less. In cross B, if you see the parent, the distance between W and M is more. So by observing this, you can tell, by observing the parents only, you can tell that which genes are linked or which genes are tightly linked and which genes are loosely linked. Then obviously, by studying the F2 generation, you will confirm that which gene is tightly linked and loosely linked. Okay. Now what Morgan found? So Morgan, he found that the genes white and yellow, they were tightly linked. They show 1.3% recombination as shown in the figure, and white and miniature wings, these are loosely linked and they show 37.2% recombination. So you can see here the white and yellow genes, they show 1.3% recombination. So in cross A, the recombinant types are only 1.3. In cross B, you can see here the genes are loosely linked. The distance between W and M is more, and you are getting recombinant types 37.2%. So white and miniature wing genes, they are loosely linked as seen in cross B, and white and yellow genes, they are loosely linked. They are closely linked. They are tightly linked as seen in cross A. So you can see in this image, this is showing you linkage. So this is a result of two dihybrid crosses conducted by Morgan. Cross A shows a crossing between gene Y and W. Cross B shows crossing between gene W and M. Here, the dominant wild type alleles are represented with positive sign. So this positive sign denotes the dominant wild type alleles. Okay, clear? Now the strength of linkage between Y and W is higher than W and M. So this we can see by analyzing this image. So let's understand this image. Now these are the parents. One is female, another one is the male. Okay, in sexual reproduction, two parents are involved. Biparental condition is there. Now, this is the F1 generation. So first of all, you will make the gametes of the parents. So this parent, this female, this will make only one type of gamete having Y and W. This male will produce two kinds of gamete. One of the gamete will contain Y plus W plus, and other will contain this Y chromosome. Okay. Then this will be the F1 generation. Okay. This chromosome Y and W, this will go with this one, and this chromosome will go with this one as well. Okay. So this chromosome can go with this one and this one, and this chromosome, and again the other chromosome can go with the other two as well. So in the F1 generation, you will get female and the male. So this one is female, this one is the male, having this Y chromosome. Okay, clear? Because this female is producing only one type of gamete having Y and W only. Males are producing two kinds of gametes here. Okay, so here this can go with this as well, this can go with this as well. So this F1 generation will be produced. Then again, you will get the F2 generation. So in the F2 generation, parental and recombinants are shown to you. Clear? So again, you can make the gametes of the female and the males. So here again, they will produce two kind of gametes. Female will produce two kind of gametes here. One with YW, another one with Y plus and W plus. And the male will also produce two kind of gametes, one with YW and another one with this Y chromosome. Now you can make again the combinations. You will get the parental type, and then you will get the recombinant type as well. So you can make recombinants. You can show recombination by crisscross method. So this Y will go with YW after recombination. So you can see Y with Y plus. Okay, this Y goes with Y plus. Clear? And you can see here the another one. This Y can go with, will go with this W plus, and this Y plus can go with W. So these are the recombinants that will be formed. Okay. So these are the recombinants and these are the parental types. Similarly, in cross B also, you should make the gametes first, then you can make the F1, then you will get the F2 generation, then you can show recombination here. So these are the recombinants. The recombinants will be W and M positive, and another one will be after recombination W positive and M. So W positive and N, and W and M positive. Okay. So basically, the thing here is to observe is that the parental combination is more in case of cross A. So here genes are tightly linked, parental combinations are, or you can say the recombinants are slightly more as compared to cross A and cross B. So here the genes are loosely linked. Now let's talk about early genetic mapping and the application. Now Sturtevant, he was a student of Morgan. He used recombination frequency to measure the distance between the genes and map their chromosomal positions. Okay, so what Sturtevant did, he used recombination frequency, also known as frequency of recombination, to measure the distance between the genes. So distance between the genes can be measured using a recombination frequency. And this was used to create genetic maps. Okay, this can be used to map the genes on the chromosomes. Now, recombination frequency. What is recombination frequency? In simple words, it is a measure of how often two genes, also known as genetic markers. So genes can be called as genetic markers as well. So how often two genes that are located on the same chromosome, they get separated during reproduction due to crossing over. So in recombination, crossing over occurs. Okay. So how often the two genes located on the same chromosome, they separate during the reproduction. Okay, during the gamete formation. Gamete formation occurs by meiosis. Now, how can you calculate recombination frequency? So there is a simple formula for that: number of recombinant progeny. divided by total number of progeny. So number of recombinant progeny is 5, total number of progeny is 10. So 5 divided by 10 into 100. So you will get here percentage recombination frequency. Okay. Now, third point, he used recombination frequency to create genetic maps. Okay. So in genetic maps, why genetic maps are made? So genetic maps, they serve as the foundation for genome sequencing as well. Okay, genetic map means mapping of the genes on the chromosomes. Or in simple word, you can say the location of the gene on the chromosome and how far a gene is located from another gene. So this can be used for genome sequencing. Okay, so genetic maps, what are genetic maps? They provide a framework for organizing and orienting DNA sequences by showing the relative positions and distances between genetic markers, means genes, along the chromosomes. Okay, so they can be, there is one chromosome, and on this chromosome, suppose there are 3 genes A, B, and C. So genetic map will tell you position of one gene relative to the other, position of A gene relative to B gene, position of A gene relative to the C gene. Okay, so this genetic maps, they are used in the genome sequencing. They are the foundation for the sequencing of the genome. Okay, and how genetic mapping can be done? By using recombination frequency. Okay, human genome sequencing project utilize genetic mapping approach. Okay, genetic mapping of the genes on the chromosome. So we'll talk about this in detail as well in the end. Okay, how recombination frequency is used to find the distance between the genes. Now let's talk about polygenic inheritance. Now, Mendel, he was studying contrasting traits. So he said flowers can be purple or they can be white. Plants can be tall or they can be dwarf. Okay. So he talked about distinct characters. But some of the traits, some trait, they show gradient or continuous variation. Okay. Like human height and skin color. There is a range of height. There is a range of skin color. So this is known as gradient or continuous variation. Example is human height. Humans are not tall or short. Okay, they show multiple variations. So humans are 5 feet, 5.1, 5.2, 5.3, 6 feet. So they show multiple variations. This is known as continuous variation or known as gradient. There is a range of phenotype. Now, what are polygenic traits then? Okay, so this polygenic trait means the traits which are controlled by 3 or more genes. Usually what is done? What is happening? One gene controls one character. One gene controls one trait. But what happens here? Poly means many and genic means genes. So when many genes control a single trait, this is known as polygenic traits, and this is known as polygenic inheritance as well. So these polygenic traits, they are controlled by 3 or more genes. Example is human height or skin color. Now, what are the characteristics of polygenic inheritance? Involves multiple genes. So multiple genes control a single character, single trait. They show influence of the environmental factors. So these polygenic traits, these traits are influenced by the environment as well. Like skin color is influenced by the environment. You go in the sunlight, then the skin color will become dark. So these polygenic characters, they are influenced by the environment as well. Another example is height. So how environment influences height? So the nutrition that you take, the exercise that you do, okay, your physical activity, okay, amount of minerals, food that you are eating, this will decide your height as well. So these polygenic traits, they are influenced by environment as well. Now, there is additive effects of the alleles here. So these traits, they are controlled by multiple genes, and each gene usually have two alleles. And these alleles show additive effect. So the overall phenotype, the final phenotype, it is due to the additive effect of each allele. So we will understand this concept. So classical example is human skin color here. Let's understand skin color inheritance now. Suppose let's take three hypothetical genes, capital A, capital B, and capital C. So the dominant alleles, capital A, capital B, capital C, they give dark skin. The recessive alleles, small a, small b, and small c, they are for light skin. Now, let's talk about genotype and phenotype correlation here. So, when all the dominant alleles are present, then what happens? The skin color will be darkest. Okay. Now, when all the recessive alleles are present, skin color will be lightest. But in the heterozygous condition, capital A, small a, capital B, small b, capital C, small c, you will get intermediate skin color. So here there is a mix of dominant and recessive alleles. So this is the additive effects of the alleles. So in this case, in the heterozygous condition, when each gene is heterozygous, intermediate skin color will appear, okay, will be observed. So now the skin color depends on number of the dominant or number of the recessive alleles. Okay. So if there was capital A, then capital A, then capital B, small b, capital C, small c, then what will happen? The skin color will be darker as compared to this intermediate one. Okay, so if there are more dominant alleles, then the skin color will be dark. If there is more of recessive alleles, then the skin color will be light. Okay, so there can be multiple combinations here. Capital A, small a, capital B, capital B, capital C, then capital A, capital B, small b, small c, small c. So there can be multiple combinations here. And the skin color and the final skin color will be dependent on the mixture of dominant and the recessive alleles. So this is known as additive effects of the allele. Now, number of dominant or recessive alleles determine the skin color intensity. Now, what are the key points to remember regarding polygenic inheritance? So they show continuous variation, means they show range of phenotypes. Multiple genes work together, and the alleles, they show additive effect. Environmental influences is significant. So these polygenic traits, they are influenced by the environment. And each allele contribute additively to the final phenotype. Another thing, these polygenic traits, they show normal distribution curve. They show bell-shaped curve here. So this is normal distribution curve or a bell-shaped curve. This is due to continuous variation. So range of phenotype is observed here. That's why you get the normal distribution curve or bell-shaped curve here. You should remember this. Now let's talk about pleiotropy now. Okay, so in polygenic inheritance, multiple genes for controlling single trait. In pleiotropy, what happens? A single gene affects multiple traits. So what is the definition of pleiotropy? A single gene is showing multiple phenotypic expression. A single gene is affecting, it exhibiting multiple phenotypic expression. Means a single gene is showing multiple phenotypes. Such genes are known as pleiotropic genes. What is the mechanism behind this pleiotropy? These genes, they usually affect the metabolic pathways. So pleiotropic genes, they affect the metabolic pathways. Clear? Now these pathways contribute to different phenotypes. So when the metabolic pathways are affected, this leads to different phenotype. This leads to multiple phenotype. So this is the mechanism behind the pleiotropy. So these pleiotropic genes, they affect the metabolic pathways, and because the metabolic pathways are affected, this leads to multiple phenotypes. Here the single gene influences multiple traits. Let's understand pleiotropy with the help of example. So we'll take the example of phenylketonuria. So this is a disease which occurs in humans. This is a metabolic disorder disease. And this is due to a mutation in single gene. So here single gene mutations occur. And because of this mutation, an enzyme is affected known as phenylalanine hydroxylase, abbreviated as PAH. So this enzyme is affected. Okay, production of this enzyme is affected. So either this enzyme is not produced, or this is produced, or this enzyme is non-functional, even if it is produced. And this enzyme is produced by a certain gene, and there is a mutation in this gene. Okay. Now, this enzyme, phenylalanine hydroxylase, what are the function and role? What are its function and role in phenylketonuria? So its main function is to convert phenylalanine to tyrosine. So these are two amino acids. These are very important amino acids in any organisms. Now, you can see if the enzyme is not produced, or if this enzyme is non-functional, then phenylalanine will not be converted into tyrosine, and phenylalanine will accumulate in the body. Okay. So this conversion from phenylalanine to tyrosine, this is very crucial for normal protein metabolism. Now, due to genetic mutations in the gene, this phenylalanine hydroxylase, this is not synthesized. So there is a deficiency of this enzyme, or this enzyme is non-functional, and this enzyme doesn't work properly. And because of this, phenylalanine accumulates in the body, and this buildup of phenylalanine leads to a condition called phenylketonuria, abbreviated as PKU. Okay. Now, because of the accumulation of phenylalanine, what happens? There are multiple effects are shown, multiple phenotypes are shown, multiple traits are observed. One of the trait is mental retardation. Another phenotype is reduced hair pigmentation, and another one is reduced skin pigmentation. So these are the three phenotypes, these are the three traits, these are the three effects which are observed in phenylketonuria. So you can see here a single gene, okay, is leading to all of these three effects. We can say the single gene is affecting three traits here. So this is known as pleiotropy. An example is phenylketonuria. This is a metabolic disorder. So these genes usually affect the metabolic pathway. So this is also a metabolic pathway where phenylalanine is being converted into tyrosine. Okay, now let's summarize the entire lecture. We talked about linkage and recombination. So what is linkage? When the genes are located close together on the same chromosome, and these genes, as they are very close to each other and they are present on the same chromosome, they are inherited together. This is known as linkage. And we also talked about recombination. This recombination occurs when the genetic material is exchanged between the chromosome during the crossing over, which occurs during the meiosis. The recombination leads to exchange of the, you can say, genetic material. Now, what are the key points related to linkage and recombination? Linked genes do not follow Mendel's law of independent assortment as observed by Morgan in an experiment with Drosophila. It didn't observe the 9:3:3:1 ratio in the F2. Now, crossing over or recombination creates new combinations as there is mixing of the genes. Then distance between the genes affect the recombination frequency. If the distance between the genes is more, then recombination frequency will be more. There will be more number of recombinants, and this recombination frequency is measured in MAP units or centimorgan. Remember this. We talked about polygenic inheritance also. So definition is traits which are controlled by multiple genes which are working together, each contributing to small effects. So there is the additive effect of these genes. Now these polygenic inheritance, okay, these polygenic traits, they show continuous variation. They are affected by environment. They show normal distribution in the populations. Bell-shaped curve is observed. Okay, and there is no clear dominance or recessive pattern. Okay, however, the genes show additive effect. Examples are human height, skin color, intelligence, and weight as well. You should remember the examples. Then we also talked about pleiotropy. Now, what is the definition? Single gene affecting multiple traits. Okay, multiple unrelated traits or characteristics. Here, one gene influences multiple phenotypic traits. Mutation can affect multiple body systems. So here, mutation occurs in a gene. Okay, and this gene, this affect multiple body systems. So this gene, this is involved maybe in the production of an enzyme, which is involved in the metabolic pathways. So basically here the metabolic pathways are affected. So gene which is being mutated, this is involved in metabolic pathways. And when this gene is mutated, then multiple effects are observed. Okay, these are important in genetic disorders because here the gene is being mutated. So this is a kind of genetic disorder. Okay, it can have both positive and negative effects. Example is phenylketonuria. Sickle cell disease is also example of pleiotropy. Now let's discuss some NCERT based question related to these topics. What is the question here? During linkage analysis of two genes A and B, okay, if the cross shows fewer recombinants, the recombinants are less than the parental types. So parental types are more. What does this indicate about the distance between these genes? So here recombinants are less, parental types are more. So distance between A and B will be more or less. What will be the answer here? So if the cross shows fewer recombinant in the parental type, this indicates that genes A and B are relatively close to each other and they are present on the same chromosome. Now there is a lower number of recombinants. This suggests that crossing over between these genes occur less frequently. And the percentage of recombinant can be used to calculate the map distance between the genes. So with the help of percentage recombination or with the help of recombination frequency, you can calculate the distance between these two genes. The closer the gene, the stronger is the linkage and lower will be the recombination frequency. Example, so this is an example of incomplete linkage because here you are getting some recombinants. In case of complete linkage, you get only parental combinations. So again, you can see in this image A and B genes, these are linked to each other because they are being inherited together. And A and C, they are not linked together because recombination is occurring between A and C. Another question, why is human height considered a classical example of polygenic inheritance? And what are the key characteristics? So what will be the answer here? Human height is considered a classic example of polygenic inheritance as the height is controlled by multiple genes. Okay, means polygenes, not just a single gene. It shows continuous variation, means it shows a range of phenotypes. And there is no distinct categories, means tall or dwarf. Shows normal distribution curve, it shows a bell-shaped curve in the population. They are influenced by environment. So height is influenced by environment such as nutrition and exercise. And each gene contributes to small additive effect to the overall phenotype. Okay, the more the number of dominant gene, more will be the height in this case. Now, no distinct dominant or recessive pattern like a monogenic inheritance here. So, in monogenic inheritance means when one gene controls a particular character, there you talk about dominance and recessive actually. But here, there is an additive effect of the alleles. So, these show normal polygenic trait, they show normal distribution curve because there is a range of phenotypes here. Next question, you have to explain pleiotropy with an example. So pleiotropy is a phenomena where single gene controls multiple seemingly unrelated phenotypic traits. One example of phenylketonuria, another example is sickle cell anemia. This show pleiotropy through a single mutation. So there is also a single base pair mutation in the hemoglobin gene that affects multiple traits. Which traits are affected? Here the red blood cells, they become sickle shaped. This is one trait. There is reduced oxygen delivery to the tissues. Then there is vision problems. Then there can be joint and bone complications as well. So you can see the mutation in here in the single gene can lead to multiple effects. So this is also example of pleiotropy. Let's discuss some keynotes that will help you memorize these concepts. So first of all, linkage can be complete linkage or incomplete linkage. Linkage was discovered by T.H. Morgan. Okay, then we talked about recombination also. So in recombination, this occurs in the prophase 1. Okay, so crossing over occurs in the prophase 1 of meiosis 1. In which stage? Pachytene stage. Okay, now you can calculate the recombination frequency, and by calculating recombination frequency, you can tell the distance between the two genes. Okay, what is the significance of recombination? It leads to genetic variation because new combinations are produced. We talked about, okay, let's talk about genetic gene mapping and centimorgans. This is very important here. What is the relationship first? 1% recombination frequency is equal to 1 map unit of 1 centimorgan. Remember this. This is very important. You can get numericals on this concept. 1% recombination frequency is equal to 1 map unit or 1 centimorgan. So this centimorgan was, you can say, was based on the scientist T.H. Morgan. Now, the higher the recombination frequency, further apart genes are on the chromosome, means the distance between the two genes is more. Now, this recombination frequency used to create genetic maps showing relative gene positions. So, genetic map, they show relative genes positions, means position of one gene relative to the other. Okay, centimorgan. So, this was named after Thomas Hunt Morgan, T.H. Morgan. This is a unit of measurement for the genetic linkage. Okay, 1 centimorgan is equal to 1% recombination frequency. Okay, clear? Helps to determine gene order and distance on the chromosomes. Okay, and using the genetic maps are being produced. Now, what are the key points here? So, there's the direct relationship. For example, if there is a 5-point recombination, so this means 5 centimorgan or 5 map unit. Okay, so question can come here. If there is a 5% recombination frequency, okay, 5% recombination between two genes, then what is the distance between two genes? It will be 5 centimorgan or 5 map units. Maximum recombination frequency is 50% in the case when the genes are totally unlinked. Remember this, maximum recombination frequency is 50%. This means maximum distance measurable is 50 centimorgan. So maximum distance between two genes is 50 centimorgan or 50 map unit. Okay, we talked about polygenic inheritance and pleiotropy as well. So polygenic inheritance means multiple genes for controlling one trait. These are the examples: kernel color in wheat. They show continuous variation. For example, human height, they show normal distribution curve. Again, example can be human skin color. Pleiotropy, one gene is affecting multiple traits. For example, phenylketonuria. So another example is sickle cell anemia, where single gene mutations occur and multiple phenotypic effects are observed. Okay, another example can be starch synthesis in the pea seeds as well. And regarding this, we have also discussed in our earlier session. Okay, so now what is the NEET focus area here? The questions can come from recombination frequency, as we have discussed here. Question can come from linkage map calculation. You should practice the numerical problems related to recombination frequency and crossing over percentages. You should understand the mechanisms behind the Morgan's experiment, and you should know how to calculate the map distance. Okay, so this will help you in the NEET exam as well. So these are some interesting questions for you. This will enhance your critical thinking ability. First question is, why do certain genes tend to be inherited together more frequently than expected by Mendel's law of independent assortment? So think over this question. Another question is related to polygenic inheritance. How multiple genes work together to produce continuous variation in human height? So why there are range of phenotypes in case of polygenic traits? Next question related to pleiotropy. Why does a single mutated gene in phenylketonuria affects multiple traits like skin color, brain development, and behavior as well? So in phenylketonuria, you see multiple traits, multiple effects are being observed. So there is a change in the skin color. Okay, brain development is affected and behavior is also affected. So why this is happening? So these are some interesting questions for you. Clear? So now we have discussed the theory. Now let's discuss the DPP related to these topics. Okay, let's start with this DPP now. First question is, in Morgan's dihybrid cross experiment on Drosophila, deviation from Mendel's 9:3 to 3:1 ratio in F2 generation was primarily due to, okay, were the genes assorting independently and they were located on different chromosomes? Next, mutation in F1 generation. Next option, physical association, means linkage of the genes on the same chromosome. Last option is environmental factors affecting phenotypic expression. What will be the answer here? So answer will be option number C here. The genes, when they are linked together, then they are inherited together. So when the genes are physically associated and when they are present on the same chromosome, then they are inherited together. Next, if two genes on the same chromosome show 1.3% recombination, what can be inferred about their linkage? Okay, the two genes, they are saying they are present on the same chromosome, they are showing only 1.3% recombination frequency. So what are the options? The genes are located far apart on the chromosome. Genes are very tightly linked. The genes follow Mendel's law of independent assortment, and the genes are located on different chromosomes. So correct answer will be option number B here. The genes are very tightly linked. That's why the recombination frequency is just 1.3%, and here the parental combination will be, you can say, more as compared to the recombination frequency. Next, Alfred Sturtman contribution to genetics was, he was a student of Morgan. What was his contribution to genetics? He discovered the structure of DNA, used recombination frequency to map the gene's position on the chromosomes. This was used for create the genetic maps. Next, proposed the law of segregation. And fourth one is identifying the role of mutations in the evolution. What would be the correct answer here? The correct answer here will be option number B. So, Sturtman, he used recombination frequency for mapping the genes on the chromosome. Next, a cross A, okay, and there is a cross B. Cross A involves gene Y and W, cross B involves gene W and M. So this experiment was conducted by Morgan. Now, which pair of genes showed higher recombination frequency? So this we have discussed in the theory. What would be the answer here? Y and W of cross A, W and M of cross B. Option number 3, both showed equal recombination, and fourth option, neither showed recombination. So what would be the correct answer here? So correct answer will be option number B. So in the cross B, the W and M were located Okay, the distance between W and M was more as compared to Y and W. So these two were loosely linked, and the number of recombinants were more as compared to cross A. Next, genetic map based on recombination frequency are particularly used for predicting the exact sequence of the gene, identifying environmental effects on the gene expression, serving as a starting point for whole genome sequencing projects, and determining the function of all genes in an organism. What will be the correct answer here? The correct answer will be option number C here, serve as a starting point for whole genome sequencing. So recombination frequencies are used to create genetic map. These genetic map can be used for sequencing of the genome. So with this, we have discussed the DPP as well. Okay, all our answers are correct. So with this, we end this session. I hope this session was quite knowledgeable for you. We'll meet you in the next session. Thank you very much for listening.